Okay, we will make a start now. So welcome everyone to our A Level AQA Biology session. Um, in today's lesson, we're going to be continuing on what we were going through in our last lesson. We started our new topic, nucleic acids. Okay, and in today's lesson, we're going to be looking at a topic from that subunit. We're going to be looking at DNA replication. Okay. Um, so if we start off now, we're going to look at semi-conservative replication of DNA. And then we're going to move on to the process because there's a process behind the semi-conservative um, replication of DNA. And then we're going to finish off by looking at some exam questions. If at all you have any questions, please let me know at any point in the Q&A because I'm here to answer them. Um, and then once we get through the topics, I will set an open Q&A if anyone has any questions, and there'll be some useful links that I post towards the end of the session. Okay, so this is what the specification says, and we are going to be covering all the points on the specification. Okay, now this part of the specification is rather simple. And the reason why it's rather simple, it's because it's not the whole process is listed on the specification. So if you just learn this process over here, word to word, the question that comes up in the exam isn't going to ask you for much more. Okay, so just learn the process behind that, and you'll be fine in the exam. Okay, so if we start off now, first of all, I'm going to go through the purpose of DNA replication. Now, you're probably wondering what the hell is DNA replication and why do we need it? Well, DNA replication, well, if we didn't have it, we wouldn't be able to grow and we wouldn't be able to repair because we saw that the DNA codes for a specific sequence of amino acids. Okay, and then once we've got that specific sequence of amino acid, then the chain of amino acids sort of fold up to form the secondary structure of a protein, then the unique 3D shape, the tertiary structure, and eventually the quaternary structure for sort of parts in the body which require more than one polypeptide chain. If we didn't have proteins, then we wouldn't have enzymes, we wouldn't be able to carry out essential biological processes within our body. So that's why DNA replication is really important. If you fall down and you've lost some blood, how is the blood going to replace the blood that you've lost? Okay, your body is always constantly growing and is constantly repairing itself. That's why we need DNA replication, basically. Okay, now, so before cells divide by mitosis or meiosis, now meiosis we do look at A level, but we look at in a different topic, which is later down in the spec. Um, Mitosis is actually in the next topic, which we look at. But before cells divide, all the DNA must be replicated to provide a copy for the new cell. So you probably remember from GCSE that when a cell is dividing, the nucleus, the internal structures of the cell must grow. So you increase the number of mitochondria in a cell, you increase the number of ribosomes in a cell, and you will already also realize that the DNA has to replicate within a cell because if the DNA doesn't replicate within a cell, a lot can happen basically. Basically, the DNA can't be transferred into the new daughter cell. So if we had a cell and this cell was to split into two different cells, if the DNA from the nucleus doesn't replicate enough, then an equal amount of DNA can't come into both daughter cells. Okay, so the DNA has to grow, repair itself, and then it is split into its two individual daughter cells. Okay, so that's the purpose of DNA replication. It forms an essential process of cell division. Okay, and a new word, a phrase that I'm going to introduce over here is that DNA replication is semi-conservative. Okay, now semi-conservative means one strand is conserved and one strand is synthesized. Okay, so we know that DNA has two strands. We looked at the two strands of DNA in our last lesson. So we saw something like this. Okay, we saw something like this, and we saw that DNA has a double helix shape. Okay, we can see a double helix there, and it consists of two strands of DNA. Okay, and we saw that as a vital comparison between the structure of RNA and the structure of DNA. Okay, so DNA has two strands. Now, think about it. If we were synthesizing two new strands of DNA once again, For example, we've got this DNA. This DNA needs to replicate. In order to replicate it, we just randomly form two new strands of DNA. It's not going to happen like that. Okay, what happens is that each of these strands will act as a template so the other strand can be synthesized. And if I was to just sort of show you here, we will go into a more detail behind this, but basically we will have each strand like this. Okay, we will have each strand like this, and then each of these strands, let's call this one, and let's call this two, will act as a template so the other strand can be formed. Okay, so this is the original DNA. Okay, we'll have over here, we have the original DNA over here. This original DNA has split, the two strands have separated, so we had one and two. Now, one will synthesize its own strand, so one will synthesize its own strand like that, and two will also synthesize their own strand like that. Okay, so now instead of one original DNA, you have two double helix strands of DNA, and this will continue to occur. So this from number one will also separate like that, and it will also separate like that. Same with number two, and it will separate. Okay, and we'll call this now number one, number two, number three, and number four. And what will happen is that each of these single strands will now form the other strand because DNA is double helix and it will form a double helix shape. Okay, that's gone wrong. Okay, the last one's gone wrong, but you get the idea that we started from this one original double-stranded piece of DNA. This has replicated over here into two strands and then into four strands. So it will keep replicating like that. And the point that I'm making is that, you know, the original strand of DNA, when we had sort of two strands like this, two strands from this original double helix, they acted as a template so that DNA replication could continue to occur. Okay, so the strands first separate and then synthesize the other strand. Okay, because DNA is double helix, it will separate, and each strand which has separated will synthesize the other strand, and that just makes sure that DNA doesn't change over time. It keeps constant, and there are no like sort of harsh changes within the DNA of different cells. Okay. So in the daughter DNA, whenever you're going through generations of cells, the daughter DNA which is produced will have one strand from the original DNA, which is newly synthesized. Okay, so that's the purpose of DNA replication. Okay, we need DNA replication. It forms a vital process within the body. Okay, now there are two enzymes which I'm just going to introduce now that are involved with DNA replication. And then we will touch more on them when we look at the process. Okay, and the two enzymes are called DNA helicase and DNA polymerase. Okay, and these two enzymes you will talk about a lot in A-level biology. um, Especially in paper one, a lot of the processes which occur in paper one will happen through DNA helicase and DNA polymerase. And also paper three, when you look at the essay, DNA and helicase and DNA polymerase are two enzymes which you talk about a lot. Okay, so these are the two enzymes that you need to have noted down, DNA helicase and DNA polymerase. Okay, we're now going to move on to the process behind DNA replication. And there are actually, there's three key steps to DNA replication that we are going to be looking at. And it's basically a process that you just need to learn. Okay, because if you learn it, then it's just going to be really easy, snappy, and ready when you come to the exam, because this topic gets assessed quite a lot. Okay, now step one, DNA helicase breaks the hydrogen bonds between the complementary base pairs between two strands within a double helix. Okay, now what does that mean? Now we looked at the structure of DNA in our last lesson. Okay, we looked at the structure of DNA, and we saw that if I just draw it out over here, let's say that this is the structure of DNA. Okay, we have the structure of DNA over here. Each strand contains a specific bond, and basically each strand contains base pairs. Okay, such as this. I've drawn them a bit bigger, but this is just to get an idea of what they are. Okay, and we looked at base pairs as in A or adenine pairs with thymine, and cytosine pairs with guanine. Okay, those are the pairs. So we have A pairs with T and C pairs with G. Okay, those are the base pairs of DNA. Now, between the two strands of DNA, these are the base pairs. So basically, the squares are the base pairs. Okay, and on one side you will have A, one side you will have T, one side you will have C, one side you will have G. Okay, and they will base pairs, and it's between these bases that hydrogen bonds occur. So between adenine and thymine, between cytosine and guanine, hydrogen bonds will be present. Okay, we saw that in our last lesson, and these hydrogen bonds ensure that the bases of the complementary base pairs are held together. Okay, when A pairs with T, okay, let's call this A and this square T. When they pair together, we don't want them to break apart. That's why the hydrogen bonds are there present between A and T to hold them in place and to hold this double helix in place. We also looked at a different bond, which was a phosphodiester bond. Um, And this is formed from the condensation reaction between the phosphate of one nucleotide and the deoxyribose of another nucleotide. Okay, and that forms the sort of single strands. Okay, so how is this single strand formed? This single strand is formed by phosphodiester bonds between many nucleotides. Okay, and the double helix, two strands come together by the force of hydrogen bonds. Okay, so DNA helicase, that's the first enzyme. DNA helicase breaks the hydrogen bonds between the complementary base pairs between two strands within a double helix. This causes the double helix to unwind. So imagine, okay, we have this original sort of shape of DNA. Okay, we have this original shape of DNA. In order for the DNA to replicate, what must happen is that this structure has to break into two single strands. Okay, it has to break into two single strands. Okay, like that. Okay, this is double stranded right now, but we want DNA to replicate. So that's why they're becoming single stranded. We're causing them to become single stranded. And how do they become single stranded? Well, over here, we had this enzyme, DNA helicase. Okay, what's holding the two strands in place? Hydrogen bonds. Hydrogen bonds are holding the two strands in place. So they're holding the two strands in place. Okay, so if we want to break the two strands and cause them to become single strands, we need to break these hydrogen bonds. And that's what DNA helicase does. DNA helicase breaks the hydrogen bonds between these complementary base pairs within the double helix. Okay, they break the bonds between these squares, causing the double helix to unwind because the hydrogen bonds have now gone, and therefore each strand will become itself, it will be by itself. Okay, so if we have a look at this image, this is normal DNA. This is normal DNA. And what's happening is that DNA helicase is coming in. And what it's doing, it's breaking these complementary base pairs. Okay, it's breaking them. So each of them will just be a normal rectangle, just a single strand, each of them will be. like that. Okay, and that's step one. Remember the key detail. You need DNA helicase breaks the hydrogen bonds between the complementary base pairs of DNA. Okay, and this causes the double helix to unwind. I.e., what's happening is that it's becoming from double-stranded into single-stranded DNA. Okay, so that's step one. Make sure you've got this detail. DNA helicase breaks hydrogen bonds between complementary base pairs of DNA. DNA helicase, hydrogen bonds, complementary base pairs of DNA. Really important phrases there. Okay, now once we've done that, so DNA helicase, we've broken the hydrogen bonds, and each double-stranded piece of DNA has now become single-stranded DNA. Okay, what next? Well, what's happening next is that each of the separated parental DNA acts as a template. That's the key phrase here. Each strand will act as a template. So originally, what we had was this. I'm going to draw again. So we had this, we had that, and then we used DNA helicase. And what we've done is we've caused it to become single-stranded. Okay, like that. So we've got from double-stranded to single-stranded. Okay, now what we're doing is that each of these single-stranded sort of pieces of DNA, one and two, they're gonna act as a template. Okay, so each will act as a template. Okay, so each of them will act as a template. Okay, and what they will do is that they will attract complementary base pairs to their strand of DNA. Okay, so over here we have a single strand of DNA, and this single strand of DNA will have base pairs attached to it. They won't be they won't be sort of attached to anyone because it's only single stranded, but they will still have base pairs on this double helix. Okay, like that. And basically, in the blood, what you have is that you have free floating base pairs of DNA. So you just have some sort of random floating base pairs just around in your blood. Okay, just like that. Okay, for example, this could be A, this could be T, this could be C, this could be G, A, T, C, G. Okay, and basically all of these squares which are present on the single-stranded DNA will also have a letter. This would be A, T, and so on. Okay, and basically this will attract a T. Okay, so a T will be attracted to here, an A will be attracted to here, a T will be attracted to here, an A will be attracted to here. Okay, and so on. So basically, over here, the double-stranded DNA has now become single-stranded. Okay, and the single-stranded DNA will attract its complementary base pairs. Okay, so on this strand, if there is an A, it will attract a T. If there's a C, it will attract a G, and vice versa. Okay, so that's basically the process that it follows. And if you wanted the information or an answer that you need, it would be this. Each of the separated parental DNA strand acts as a template. So the double-stranded DNA has become single-stranded, and the single-stranded DNA is acting as a template. Okay, and then free-floating nucleotides, i.e. these bases, free-floating nucleotides, these squares that I've drawn, they will be attracted to the complementary base pairs on the template strand of DNA. Okay, so the template strand of DNA has some exposed bases. They will attract the complementary base pairs, which are just floating around near the single-stranded DNA. Okay, now if we'll have they won't join together, they will just sort of attract. Okay, so over here, if we have a look at this diagram, these are our free floating nucleotides. So we have our free floating nucleotides. And what's happening is that these free-floating nucleotides will base free-floating nucleotides will be attracted to the exposed bases on the template strands of DNA. So this is template strand 1, this is template strand 2. As you can see, the bases are exposed. So bases are exposed, and these free-floating nucleotides will be attracted to the exposed bases. Okay, that's basically what's going to happen. Okay. So if we put that into sort of word, so each of the separated parental DNA acts as a template. Free-floating nucleotides in the nucleus are attracted to the complementary base pairs on the template strand of parental DNA, not joined together. That is step 2. So step 1 is that this double-stranded DNA becomes single-stranded. Okay, so it goes from double-stranded to single-stranded, just like that. And then once it's become single-stranded, the exposed sort of bases which are present on the single strand will attract the complementary base pairs which are just floating around. Okay, and the reason why it does that is because each single strand is acting as a template essentially. Okay, now the final step, there is another step after this, but it's just a summary step. This is the final step. So remember, there were two enzymes. There were DNA helicase and there was DNA polymerase. Okay, we've talked about DNA helicase already. DNA polymerase. Basically, now the free-floating nucleotides are attached, they're basically attracted to the exposed base pairs on the template strand. How are those nucleotides going to join together? Okay, they have to join together via an enzyme. Okay, and it's DNA polymerase that catalyzes the joining of adjacent nucleotides. So over here, we had these exposed bases. They were attracted to the bases which are on the um, template strands. Okay, so we had, for example, this and this. Okay, and we had sort of exposed bases like that, like this, okay? And we had some free floating nucleotides just going around, okay, just like that. And we saw that A pairs with T and C pairs with G. And basically what's happening over here, so basically what's happening here, for example, A will go pair with T, C will go pair with G, and so on. They will just be attracted to each other. Okay, so you will end up forming sort of something like this. Okay, something like that. Okay, they're not actually joined together, but we need them to join together. And that's what DNA polymerase is there to do. Okay, so DNA polymerase is basically there to join, you know, the attracted bases form this bond. Okay, they need to attract, they're attracted to each other. DNA polymerase will catalyze. Okay, it will speed up the joining of the bases together. Okay, so the adjacent nucleotides are joined together. DNA polymerase catalyzes the joining of adjacent nucleotides. Okay, that's step 3. So we saw in step 1, DNA helicase breaks the adjacent nucleotides, breaks the hydrogen bonds between the adjacent nucleotides. And then we saw in step 2 that each will act as a template. And then in step 3, what you need to know is that DNA polymerase will catalyze the joining of the adjacent nucleotides. And then basically, if there was a step 4, I've just put it as finally 2 sets of daughter DNA, this over here, containing one strand of parental DNA and one newly synthesized strand is formed. Okay, so we had this over here. Originally, basically what that is saying is that one strand is from our original parent. Okay, only one strand is synthesized in each helix, in each double-stranded bit of DNA. One is the new strand, one is old. Okay, so one is old, one is new, basically. Okay, and that's basically the idea of semi-conservative replication of DNA. Okay, so we started with DNA helicase, template strand of DNA, DNA polymerase, and then semi-conservative replication. The final replicated DNA has one original strand of DNA and one synthesized strand of DNA. And that way, DNA will always be conserved. As we go down, it can replicate as many times as it wants, and one strand will always be conserved. So you are basically holding the same DNA each time, and different cells will also have the same DNA. They won't have some random DNA within your body. Okay. Shortened version to remember. I remembered it like this whenever I was remembering the processes, because there were exam questions which were literally asking me to list the process. So step one, DNA helicase. Step two, DNA template. Step three, DNA polymerase, condensation phosphodiester bond. And then step four, final DNA polymer is produced. Okay, those were the shortened steps that I remembered the process with.